Club. As usual, we have to have a technical issue before we start. So that's tradition, I think, by now. So today we have Junior, who's a method assistant in the lab of Andreas Poland. Thank you for accepting to give this talk. And over to you, Andreas, to introduce Junior.
Yeah, thank you. So it's my pleasure to introduce June. June started 2019 in June, actually, I think June, July, you started in the group.
Yeah, at the end of July.
Yeah. So it's about 3 years and 10 months or so. He did his master's at Zhengzhou University in China, of course, before he moved then for the PhD to the Institute of Nutrition and Health at Shanghai University, which is also an institute of the Chinese Academy of Science. And during his PhD, he worked on metabolic enzymes, but he had quite a challenging example because no matter what he grew with the crystals, they were kind of twinned or pseudo-translated. So I was thinking, okay, the biochemistry seems to be fine because otherwise you wouldn't get crystals. But then he had actually to use computational methods to remove the diffraction spots of the crystal that he didn't want to analyze and then get a clean data set to solve the structure. So when he asked to join our group at that time, I thought, if he can process crystallography data like this, he will also be able to process EM data. So that was a very good start for us. And then he came to work, he immediately said, I would like to work on Separase. So I had to think about a project that was related to this enzyme that is involved in chromosome segregation. And after a bit of a slow start where we still tried to do some crystallography work together, the pandemic actually hit and June had time to sit at home and process the data that existed already. And every day it got a bit better. And I thought, like, if he continues like this, in 2 weeks, we will have a real new structure. And it really was like that. So we could then go and collect some data at the university, at the Max Planck Institute in Dortmund, which gave us the final data set to solve the structure. When the first structure was solved, he had already the biochemistry for the second complex established, and we could collect more data. So things became much more fluent after a time. So the first half a year was a little bit slow, but then you picked up pace quite quickly. And I was very happy that our project worked so well after that initial hiccup. And then now he has been moved from a postdoc position to a maître assistant position since one month. And so now the floor is yours to tell us all about what Separase is doing in chromosome segregation.
Yeah, thanks a lot, Andreas. Okay, no, we just start directly. Yes, as Andreas introduced, I'm now a senior scientist in the Poland lab. Today we share my work on the human separase. I mean, in the past 3 years, roughly, it's about human separase regulating the chromosome segregation, mainly about the separase inhibition and the substrate recognition. Actually, the cell division, I think all of us will know this, how it goes. In one sentence, simple sentence, that cell division is a process dividing the duplicated genetic materials into 2 daughter cells equally, as well as the cellular components. And after the DNA duplication, the cell will enter the mitosis. It consisting of 4 phases: prophase, metaphase, anaphase, and lastly, the cytokinesis. Specifically, in the metaphase, the old chromosomes are well aligned at the mitotic spindle plates, and all of them are well attached to the microtubules, but interacting of the outer kinetic core with the macrotubules. The inner kinetic core interacts with the centromeric DNA, which has a special histone, can distinguish from the normal nucleosome. And in the centromere, the coenzyme ring, which is a protein complex ring, can hold the 2 sister chromatids together. The coercion ring normally has the four proteins. SMC1 and 3 can form a dimer through the hinge domain, and the head domain actually is the adipase. Together with the DNA loader, it can load the DNA into the coercion ring. SEC1 can bind to SMC1, 3, and SE1 or 2 at the same time. SE1 or 2 actually is a regulatory subunit, which is believed has a function in the deloading and the coercive movement on the chromosome arms. During the metaphase to anaphase transition, there's one important mechanism called spindle assembly checkpoint mechanism. To make sure that all the chromosomes are well attached on the microtubules. But if there's errors in the attachment or there's not all the chromosomes are well attached, the SAC signal will be on. It will produce the effector MCC complex. It means the mitotic checkpoint complex. It will bind to the anaphase promoting complex, which is a giant E3 ligase, and inhibits the activity. However, when all the chromosomes are well attached, the SAC signal mechanism will be off, and it will make the anaphase promoting complex be active. It will catalyze the ubiquitination of the universal inhibitor of separase on the N-terminus. The ubiquitination of securin will result in the destruction of securin through the protease system and releasing the active separase. In the human, there's another inhibitory complex, is the separase binding to CDK1 cyclin B1. This complex formation depends on the phosphorylation of serine 1126 on separase. Actually, similar to the securin destruction, cyclin B1 destruction also requires the ubiquitination of N-terminus by the APC. Once separase is activated, then it will recognize SEC1 at the EIMR cleaved site, and it cleave SEC1 and open the coercion ring, releasing the two sister chromatids. Then the two chromosomes will pull to the opposite side of the dividing cells. Then what is separase? Actually, human separase is a quite large protein. It is actually the cysteine protease. It has 3 domains. The active sites consist of histidine and cysteine located at the very C-terminal of separase. In the TPR-like domain, there are 2 very important flexible loops called insert 1 and insert 2. They have multi-binding sites for separase binding partners. For example, in insert 1, there is a serine 1126 can be phosphorylated by the CDK1 cyclin B1, which is responsible for the cyclin B1 binding. In the insert 2, there is a PP2A binding motif and a CDC6-like motif, which is responsible for binding CDK1. And also insert 2 has the 3 cleavage sites. Actually, after cleavage, the 2 fragments are still stick together, but not affecting the activity of separase itself. Human separase actually have multiple substrates, but one of the most important one is SEC1. SEC1 is a quite flexible protein. It has like multiple regions responsible binding for SMC1 and 3, and also SE1 or 2. It has 2 cleavage sites, and also previous studies have identified that the ALP motif is quite important for the SEC1 cleavage by the separase. Actually, the cleavage site is quite conserved from the yeast to human. Separase will recognize this motif, EXXR motif, and cleave just after the arginine. Now we know that the separase is highly regulated in the pathways, and then we are wondering firstly, how separase is inhibited by its inhibitors. And secondly, after the separase is activated, it will cleave itself. So we are interested in how this separase recognizes the autocleavage sites. And lastly, and importantly, we want to know that how separase recognize its substrates, SEC1 or the wholly covalent complex. So the first part of my talk is about the inhibition separase firstly. But before I start, thanks a lot. I have to give many thanks to our collaborators, Chloe in Devin Morgan's lab, and Pierre, our former lab member, and also Andreas. Firstly, we co-expressed and purified the separase securing complex, and also for the separase CCC complex, we expressed them individually and incubated them together with the ADP in vitro to assemble the wholly complex. We applied the samples with onto the graphene oxide grids and connected data with the electron microscopy. And finally, we get the three instructions of the separase securing complex at 2.9 angstrom, and also the separase CCC complex at 3.6 angstrom. Okay. Let's see the overall. Sorry. Overall in no. Okay, there's a movie here actually. Let's see the overall texture of the two inhibitory complexes, how they look like. As you see, actually the binding modes is quite different of the two inhibitory complexes. With the separase securing complex, with the help with AlphaFold prediction, we built a composition model consistent of the full length of separase and the fragments of securing binding. We see that securing actually binds to separase at all the three domains from N term to C term. This binding mode is similar to the other species actually. We see the securing binding to the older domains of separase, specifically at the SPD domain, securing binding across the active site, indicating that securing probably acts as a pseudo substrate to inhibit activity. And also in the human securing, we found several motifs and a hydrophobic fragment mediating the interaction. Then we think to delete these elements in securing to see if securing can still binds to separase. Then with the pull down experiment, we surprisingly we see that once the hydrophobic fragment is removed, securing cannot bind separase anymore. Securing cannot bind separase anymore, indicating that this hydrophobic fragment is quite important. Constantly, once this fragment is removed, securing cannot inhibit the activity of separase anymore. But that's actually, you may wonder that because securing still have the three domains left, but the interaction the interaction cannot be detected anymore. That's actually a question I will explain later. When I checked the structure carefully, we found that this hydrophobic fragment mediating a lot extensively hydrophobic interactions actually. You see the securin go through a hydrophobic channel on separase. With the CCC bonds to separase, the structure is completely different, and the binding mode is also different. The CCC mainly interacts with the separase through the TPR-like domain. Specifically, the cyclin B binding loop from insert 1 wraps around cyclin B1 but with the serine 1126 at its center. At the same time, the fragment from insert 2 bind separate itself and also the CDK1 at the same time. We call this fragment binding to separase with the auto-inhibitor loop 3. At the same time, there's another loop called A1 bind also becomes ordered and binding next to the active site separate. So next we compare the two modes of separase inhibition by securin and the CCC complex. We see in the securin, the three motifs exist in the securin itself and also in the SEC1. So securin actually has multiple motifs, multiple pseudo-substrate motifs in inhibiting the activity of separase. However, in the separate CCC complex, this motif exists located at the separate itself. Like, for example, A1 has the NXRX5 mode, 5E motif, 5 is the hydrophobic residue. And the AO3 has the NFS sequence, which mimicking the EIE 5XX5 motif interaction. And lastly, that's maybe not so important. We see that cyclin B1 binding loop binds to the space between the L3 and L1, and further to occlude the SEC1 binding. When we check the interactions in details, actually, this is too much details here. We see all the motifs binding to separated them is quite similar. So next we checked how important is motifs in the cleavage of SEC1 by separase. We just mutated the NX, LX, 5E motif and LP motif for the important residues into alanine and check the activity. We see that once these motifs are mutated, the cleavage of SEC1 is completely abolished. And next we check the auto-inhibitor loops in the cleavage of SEC1 by separase. A1 delta is just remove the A1 loop. L3 we have three mutants. Either we remove the whole insert 2, or either we just remove L3, or either we just remove the species like 6-line motif plus the L3. And test all the activity of the mutants of separase. We found once we removed L1 loop and also L3 loop, the activity of separase increased by around 2-fold. But except the insert 2 deletion. For now, we still have no, obvious ideas how removing the whole insert 2 affect the activity of separate itself. Apparently, it has the same activity as wild type. So as a summary, we see here with a brief summary of the inhibition of the securin and the CCC. Securin and the CCC, they are using the pseudo-substrate motifs to inhibit the activity of separase. In the securin, these motifs located in the securin itself. However, in the CCC separate complex, these motifs located at separate itself, like A1, A3. And in the separate, actually, there's two new findings in the separate CCC structures. I won't tell too much. I won't tell too much details because if you're interested in this cyclin B1 phosphorylated binding pocket and the inhibition of the CDK1 by the separase, we can talk afterwards. And now we know that the SCC1 has motifs, which is quite important for the cleavage by separase. So we are wondering that how separase can recognize this motif in SCC1. And also previous studies show that the linker between these motifs are also quite important for the cleavage. So we also want to know how this linker binding to separase and mediating the interactions. But before the substrate human, before the separase substrate structure, I want to talk about the separase in apo state, because this is a quite important finding. In the previous study, there are two methods to obtain the apo separase. Actually, the first one involves either purified separase securing complex and then incubate the complex with the egg extract. Because in egg extract, there's the APC proteasome system. It will degrade securin and release the active separase. In 2019, David Morgan's lab developed a new method using the bacteria protease to degrade the securin. What they did is that they fused a CLPXP protease diagram sequence at the N-term securin. And this sequence will be recognized by the protease system with the pholdase and the translocase and the peptide days. Securin will gradually degraded by this proteasome and then releasing the active separase. So actually, the two methods requires the proteasome. We are wondering that if we can use the easier way to obtain the apo separase. Before, we use the short securin fused the N-terminal separase to make sure that separase is soluble and expressed well. So what happens if we just shorten this fused securin to like 10 amino acid at the N-term? When we test the expression, we found that it actually is soluble expressed and it can be purified. Then next, I was thinking maybe we just remove the securin at the N-term, just express separase itself. Can we get the protein or not? Then indeed, we can purify the separase without any securin fused. And together with the Makita in our lab postdoc, we purified the active separase. We can see the outer cleavage of separase itself. It has multiple fragments, indicating that separase expressed without any securin fused is still active. Then next, we just apply these two samples on the grid and the kinetic data, and actually on our Actica, and obtain the high resolution structures of the two apo separates. I mean, like 3.1 and 3.2, which is clear enough for us to build a model. Okay, no, I just skip. Then, okay, then what do we find in this apo separase? Surprisingly, first we found that the A1 still binding next to the active site, which is actually surprised us or not, because in the CCC separase complex, A1 is binding there. But for now, without CCC, A1 is still binding here. And secondly, we identified the densities for the outer cleavage sequence binding to the exact active site and the other binding sites. So then actually the modeling of the outer cleavage sequence has a trial and errors, because the density actually is not so, I mean, 100% sure. But with when we see the active aposeparase, we only see the A1 binding, but not the autocleavage side binding, because it's already been removed. So we are more sure that what we identified of the sequence here actually belongs to the autocleavage sites. So the aposeparase, then I just build a model of the dead aposeparase with the L1 and the autocleavage sites model. We see that the autocleavage sites bind into active autocleavage sites bind into the active site and also the other binding sites of separase. Then we are thinking that maybe this binding mode of autocleavage sites to separase is similar to the SEC1 binding to separase. I just did a alpha42 prediction to see if the binding is similar or not. As you see, actually, quite impressive that the AlphaFold predicted roughly the same size of binding on separase. When compared of the two structures, I mean the predicted one and also the aposeparase, we see the binding path of SEC1 and autocleavage site, they're the same, almost the same. Now, here's a brief summary of the aposeparase. I mean, in the active and the dead aposeparase, we see the L1 binding next to the active site. And also in the dead separase, we also see the autocleavage site binding to the active site and the other side and the extra side. So, and because we are trying a lot to obtain the separate SEC1 complex, so now we know that the autocleavage site probably an L1 loop probably will interfere SEC1 binding. So next step. The third part is about the substrate recognition by human separase. We have a strategy, try to obtain the SEC1 bind to separase and to obtain complex and resolve the structure. But here we first just remove the autocleavage sites and then fuse the SEC1 to N-term separase to make sure that the SEC1 is always together with separase to increase the chance to see the binding of SEC1 to separase in the structure. And also, we purified this fusion protein, even though we have contaminations and degradations, we still connect data on this fusion protein. Then what we see in our reconstructions, actually, as you see, we only see the L1 binding to the active site, but not the SEC1 binding. This is a bit disappointing at the first when we see the 3D reconstruction. Okay. Then we are thinking what will happen if we just also remove the L1 loop, because we see with the aposeparase and also separate with the SEC1 fusion structure, we always see L1 binding there. So maybe A1 is the important factor affecting the binding of SEC1 to separase. So I just test the two enzymes. One enzyme is the wild type. Two enzyme is the separase removing A1 loop. As you see, actually, once the A1 is removed, the activity of separase is increased a lot. And then I think actually it's same for the pull down experiment. Here I mutated the active site of separase make the separate intact on the gel. So once L1 is removed and also other clip sites removed, I can see the pull down bands of separase only at the 50 millimolar salt. It means that the SEC1 binding separase is also the salt dependent. So next, we know that the L1 and other clip sites really interfere the SEC1 binding. So we just remove L1 and also replacing the other clip sites as SEC1 fragments. To make sure this time, make sure to see if we can see the SEC1 binding to separase in the structure. And then just last week we connect some data and obtained around 4 angstrom reconstructions of this fusion protein. Indeed, actually, even though electron density is not so strong, we still see the SEC1 binding there. It's then when I just use this map to build a model, even though the residues of SEC1 is not assigned because I cannot because density is not so well resolved. I cannot build atomic model of SEC1. We see the binding of the SEC1 just across the across the active site and also the extra site of separase. Then I compare the aposeparase and the substrate SEC1 binding to separase. We see that indeed the binding modes of the autoclavity sites and the SEC1 they are quite similar, and also the SEC1 binding site is also similar to L1 binding. Means that L1 really interfered the SEC1 binding. When I compare the securing separase and the SEC1 separase structures, we see in the SP domain, the binding path is quite similar. But after the LP motif here, we see the binding path of the SEC1 and securing is different and is different. And also, surprisingly, we found that the L2 loop, I didn't mention it so much before, but here it's quite interesting. We see the L2 loop has a conformational change of the two in the two complexes. In the separate securing complex, we see L2 binding at A position. As you see, apparently this position will clash with SEC1 binding, which prevent SEC1 binding to separase. In the SEC1 separate structure, we see the loop has a conformational change to the B position. Actually, will prevent as securing binding. As you see, actually, the separase has involved multiple mechanisms to make sure that SEC1 is cleaved at the right time and the right position. And lastly, previous studies show that the separase can localize on chromosomes from the metaphase, from the prophase even to the metaphase, but disappeared at anaphase. With the detailed localization, we found that the separase localized at the centromere. People have tested the interaction between the DNA and the separase, and with the 50 BP DNA, the affinity is roughly 200 nanomolar, and also the binding affinity depends on the length of the DNA. With the short peptide as substrate, they tested the DNA stimulation on the activity of separase. We found that there's actually no big differences. It means that DNA itself cannot stimulate separase activity. However, when they use the coercin as a substrate and plus DNA, they found that the stimulation of DNA is quite impressive. With DNA, the activity of Separase actually can increase by 100-fold. And also this stimulation is depends on the DNA length. So our last aim, I mean, actually it's my dream to get the coercion separase and together with DNA complex to see how the DNA and also the other components of coercion, how they stimulate the activity of separase. But now, actually, we tried a lot with this, complex formation. Until we got the aposeparase, we know that the L1 and autoclavicide will affect the binding of SEC1 separase. So next, I will try just remove this two L1 and autoclavicides and just mix the coercion complex and the separate protein and DNA together to see if we can obtain the whole complex. If we obtain the complex, I think we try to get the structure and to see some new findings there. And lastly, I have to thank to my lab members and Andreas, my supervisor, and other lab members, and also our collaborators from UCSF, Morgan, Chloe, and also from MPI, Dortmund, Stefan, and Toby, and also the David Buffer from MRC, LMB, and also our former first manager, Yasha, and the first manager in the FMI, Zimo, because they did the data connections for us. Okay, that's all. Thank you. Thanks for the attention.
Thank you so much, Emmanuel. Even for someone who doesn't understand lots EM data, you made it very accessible and very nice. I really enjoyed it. So, questions for Junior?
Hi, beautiful talk. So I'm curious about the phosphatase in the future. Do you have any plans to look at the phosphatase binding that you said there were PP2A binding site?
You mean the second B1 binding site, second B1, right?
No, I was thinking PP2A.
PP2A.
Yeah, in the future. No.
Okay. Actually, the P2A binding to separate is mediated by it actually have overlap with the autoclavicides. Once the separase is cleaved, PP2A binding is abolished. So for now, the dephosphorylation of serine 1126 PP2A is not, for now, it's not validated by us yet, right? So we are not sure that PP2A is responsible for the dephosphorylation of this serine 1126. Maybe you can try first the activity assays to make sure that PP2A is the enzyme to do this job, and then next we will resolve the structure of PP2A binding phosphorylated separase to see how the phosphate can recognize by the PP2A. This project has established by the Sophia in our lab. So this is our future plans.
Well, at the end of the talk, we spoke about the future, right? The coenzyme ring and the DNA of the separase.
Yeah.
And as I understood, you said that you want to use the separase without the outing group 1 and the
Clavicide.
yeah, the clavicide. So it's gonna be an active separase, right?
Be inactive actually.
It's the inactive.
Yeah, because
If it was active, my question is like, aren't you just afraid that you're not gonna see anything of the freaks because it's active?
yeah, yes, because we still want to see the arginine from the substrate insert into the pocket of the active site. So we want to keep this, yeah, so we use the inactive separase, yeah.
This has been almost like a group meeting also for me because a lot of the results I haven't seen before on this group.
Or
So how can you go with your salt concentration? Because this pull down was also new for me. You show that with 50 millimolar salt, you see a band of separase. But what happens if you go to 20 millimolar salt or 10 millimolar salt? And are you sure that the separase is still soluble, or maybe just precipitates during the pull down? Or
yeah, for the 50 millimolar salt, I'm sure that is soluble. Even because I diluted the separase with the 50 millimolar salt, is still alive. So I never tested below the 50 millimolar salt. Maybe it's still working at like 40 or 30. Probably it work. I don't know. Maybe so we need to try. And here, actually, here I use an MVP pull down, bead pull down, and here's just illusion. So
it's not from the beads?
yeah, not from beads. So it means that the soluble, yeah, is soluble.
But have you ever put your protein on the bench overnight at different salt concentration and see if you can see a pellet the next morning or something like that?
No, this
10,
and
20, 40, 50 millimolar salt, if you compare.
this I never tried. So it should. I think even with the 50 millimolar salt overnight, maybe it will be a problem. Probably it will. I have like similar experiments before, so I have a feeling it won't. Be alive overnight. Yeah. Actually, I haven't mentioned it because we see. Okay. Actually, yeah. With this pull down, we see that security, even though it has multiple motifs binding to separase, right? But in the pull down, we didn't detect this interaction. That's at the beginning we thought it's very weird because we see a lot of interactions in the structure. But here, because I used the dead separase, so it means that the autocleavage site is there and also A1 is there. So apparently it will affect the binding of securine to separase. Now we explain from structure, we explain what we observed, which is not normal.
Yes, all pizzas arrive from there. So we can take this discussion at the pizzas, and thank you again, Junio, for accepting this talk. Yeah, we can have. It was actually Javier who invited you today. Javier.